Thank you all for being here. My name is Preeti Kayastha, and today I will be defending my study, which is titled Temporal Transcriptome Profile Reveals A's and 6 Specific Signatures of Capsaicin Intervention in Drosophila. Aging is defined as this gradual decline in physiological function that is accompanied with the increased risk of molecular disorder, immune dysfunction, and decreased stress resistance. As a body ages, there are many changes in multiple levels. These changes can be summarized into 12 hallmarks. The hallmarks include genome instability, telomere attrition, epigenetic alteration, loss of proteostasis, disabled macroautophagy that all belong to primary hallmarks. The antagonistic hallmarks include cellular senescence, mitochondrial dysfunction, and deregulated nutrient sensing. And the integrative hallmarks are dysbiosis, inflammation, altered intercellular communication, and stem cell exhaustion. Now, why is it important to study this issue of aging? We know that the Asian population is increasing, which means that we might be heading towards a future with more number of aged population with less younger population to take care of them. With aging comes multiple aging-related disorders and diseases such as Alzheimer, Parkinson, and many more. This necessitates the need to create a sustainable healthcare system so that we can have an improved quality of life even in older age. So now the question is not just to extend the lifespan. It is also equally important to improve the health span when we age as well. Now there are dietary interventions that helps in improving this lifespan as well as health span in organisms. The dietary intervention also includes phytochemicals such as rapamycin, resveratrol, curcumin, and many more. Among them, the one that we're working with is capsaicin. So capsaicin is this vanillyl alkaloid that is naturally found in peppers of the genus Capsicum. In the mammals, these capsaicins they bind in TRPV1 receptor, the transient receptor potential vanilloid 1. This results in flow of the ions, which is the reason why we feel the burning sensation whenever we consume the peppers. Through the TRPV1 dependent mechanism, there are multiple beneficial in our body, such as antioxidant, antimicrobial, gastroprotective, and even cancer prevention, and many more. But what about those effects that happen independent to TRPV1? Now, there are studies that have explored these effects of TRPV1 independent mechanism, but those are mostly limited only in cell culture system or TRPV1 knockout models. Very few have worked with an organism without a TRPV1 ortholog or non-existent TRPV1. So that is why we are working with this model organism Drosophila. It does not have the ortholog for human TRPV1. It also shares longevity pathways with humans, and there are.
Someone is just click at me. So must be with them. Yeah.
So there are shared longevity pathways of Drosophila with humans. We also share 60% of genetic similarity with the flies. Similarly, as the genome is well characterized, it is also easy for genetic manipulation, which will be helpful for future functional analysis. It has a short lifespan, which means the aging-related experiments can be conducted in a short span of time. It is also easy to maintain the dietary control and environmental consistency, which makes it suitable for our study. Aging in Drosophila involves progressive decline across multiple systems. In the brain, the mRNA level declines and disrupts the gene regulatory network. In the muscles, there is loss of proteostasis and impaired muscular function leading to systemic muscular aging. In the gut, there is imbalanced microbiota which causes immune dysfunction and reduced lifespan. In the fat body of flies, there is increment in the antimicrobial peptide, hence leading to chronic responses. Also, in the heart, the myocardia stiffens as the fly ages, and there is also arrhythmia. So we can see that at the multiple levels of brain, muscles, gut, fat body, and myocardia, we see that a fly has multiple effect in its multiple systems as it ages. So these are the major two reasons why we are studying capsaicin in Drosophila. Firstly, as I mentioned earlier, we are trying to understand the TRPV1 independent beneficial effects in the flies. Next is a previous study from our lab by Gerardo Flores Iga showed that flies showed sensory behavior towards capsaicin. So we are also trying to understand what could be the physiological effect behind it. So my overall objective is to determine whether capsaicin can mitigate aging-associated decline in Drosophila, assessed at multiple time points in both the sexes. The specific objectives include, first, to observe if capsaicin can extend lifespan in Drosophila. Second, to perform physiological assays to test whether capsaicin preserves function and resilience. Third, to understand the molecular basis of capsaicin's effects by investigating how it modulates transcriptomic profile. Now, this is how the study was designed. All the experiments were conducted in W1118 flies. The flies were sorted males and females into males and females, and then they were exposed in either one of the two diets, either the control diet or a treatment diet of capsaicin 2 millimolar concentration. The reason we went ahead with this concentration was because the previous study from our lab found the sensory behavior of flies in this concentration. So we went ahead with this. Now, the experimental plan can be divided into 3 panels based on addressing each objective. Our first objective is to conduct a survival analysis. For that, the flies were exposed to one of the two diets, either control or the capsaicin, and then mortality was recorded every 2 to 3 days. Then we plotted a Kaplan-Meier survival curve comparing flies in control diet versus the capsaicin diet. Second, we performed these assays quantifying the physiological effect of capsaicin. We looked at body weight measurement, negative geotaxis assay, negative geotaxis assay, cold stress resistance, and starvation stress resistance assay. Now, instead of looking at a single time point, we looked these effects at multiple time points because we were interested in observing the effect of capsaicin in a temporal perspective. So in each time points, they were collected and these experiments were conducted. Now, these time points represent different stages of adult life of Drosophila. Day 2 represents the newly closed, day 12, young adults, day 30, early onset of aging, and day 50, late aging. Following the similar experimental design, we also profiled the transcriptome in these respective time points. So we collected the flies, extracted RNA, and sent for the sequencing. And then the sequence results were used for analysis, such as volcanoplot for observing the differentially expressed genes, dot plot showing pathways or gene ontology through overrepresentation analysis or gene set enrichment analysis, heat map showing DEC, and we also performed WZ-CNA analysis, weighted gene co-expression network analysis to see a coordinated expression network. Now, let's look at the results and start with survival analysis. As I mentioned earlier, we had compared the survival curve between capsaicin-fed flies and control-fed flies. And what we observed that in both males and females, they both reported significantly living longer when they were fed with capsaicin diet as compared to the control diet. In females, there was a mean lifespan extension of 16.56%, whereas in case of males, the lifespan extension was 12.91% in the males. These results are consistent with previously published studies of Drosophila capsaicin extending lifespan in Drosophila. Next, we perform the physiological assays, as mentioned earlier. The first one that we see was the climbing assay. So in the climbing assay, we tap the flies so that they settle at the bottom. And because flies have this negative geotaxis, they always climb upwards against gravity. So we use that to quantify the climbing performance. These experiments were conducted at all the time points, as mentioned earlier. And here we saw that in the earlier time points of day 12 and day 30, there was no any significant difference between the climbing performance of flies in females. But in the old females of day 50, the capsaicin-fed flies had improved climbing performance than the compared one. We looked at the climbing performance at males also, but did not report any significant difference. Excuse me. Next, we looked at the stress resistance, starting with the starvation stress. So instead of providing the flies with the normal Bloomington diet with nutrition, we provided them the other diet, whose only purpose was to provide them hydration and no nutrition. Again, comparing them at all the time points, we saw that in female at day 30, the flies fed with the capsaicin, they had increased survivability as compared to the control ones. This significant difference was not observed in males. I had also compared them in the earlier time point of day 12, and I observed that in day 12, control diet had improved improved starvation survival probability than the capsaicin one. Again, no any significant difference in the male at day 12 and also at day 50 as well. Usually, these changes in starvation stress is accompanied due to changes in the body weight. So we next compare the body weight between capsaicin-fed and control-fed flies in both males and females at different time points, and we did not observe any significant difference, which suggests that the changes that we are observing with starvation stress resistance are independent to changes in the body weight. Next assay we observed was cold stress. So in the time points, we collected the flies. We exposed them the cool shock by keeping them in 4 degrees Celsius for 14 hours. And then they were taken out in the normal temperature, 25 degrees Celsius, and allowed to recover for 25 hours. And then we measured the survival percentage of the flies. While doing so, we found that in the middle age of day 30, female flies showed improved survival percentage after they were induced to cold stress. That was significant, but in males, there was no any significant differences. Again, looking at other time points, there was no any significant difference. This shows a female-specific physiological effect or benefit from capsaicin. Now, let's move on to our transcriptomic results. As I said earlier, these are the analysis that we did. The first analysis I did with the transcriptomic was the principal component analysis. Here, I saw PC1 had the highest. PC1 had the high variance of 95%, and the samples were clustering based on sexes. As we can see that these female samples are clustering to the left and the males to the right. Since sex was such a dominant driver of transcriptional variation in Drosophila, even with the capsaicin intervention, the differential expression analysis from here onwards were conducted separately in males and females. After this, we also looked at the PCA within the sexes, where we saw that after sex now, Ace was the dominant variance, as we see the day 12 samples clustering here to the left, followed by, day 2 sample clustering here to the left, followed by day 12, day 30, and day 50. A similar clustering was observed in males as well, where ace-dependent clustering was found after the sex. Then I also plotted a three-way factorial ANOVA, and here I observed that sex is significant for the largest set of genes, which is 11,000. This is more than 90% of genes that we observed over here, and then followed by age. So here, because it was a three-way factorial ANOVA, we looked at the ANOVA with the main effect of sex, age, and treatment, and also the interaction between the two-way and three-way, such as treatment age, sex treatment age. Looking at the main effect of treatment, we observed only 15 genes being significant, which did suggest that capsaicin might be having a very negligible effect overall. But then when we look at its interaction with other terms like age and sex, we did see more significant genes, which suggests that capsaicin might not act as a main effect, but rather through interaction with sex and age, and it might be heavily context dependent. Next, we look at the number of genes significant. We looked at the genes that are significantly differentially expressed with a nominal p-value of less than 0.05. These red bars show the upregulated genes, and the blue bar show the downregulated genes. Now we computed the number of DEX for all the time points in both female as well as male. And here we see that female has the highest number of DEX at day 12, as there were 2,355 upregulated genes and 1975 downregulated DEX. Then the number of DEX decreased in day 30, as only 1455 upregulated and 1427 downregulated DEX were observed. Day 50 had the least number of DEX, as 446 were upregulated and 61 downregulated. The pattern was a bit different in case of males, because in the day 12, they had the least number of upregulated DEX with 492, and there were 762 downregulated DEX. Day 30, the number of DEX increased by 1104 in upregulation and 1465 in downregulation. Day 50 had the highest number of upregulated DEX with 1410, but also had the lowest number of DEX at 400. So females and males had a different response to capsaicin just based on the number of DEX being upregulated at that particular time point. Now let's look at the differential expression and pathway enrichment individually to these conditions. So in the female day 12, we see upregulation of genes such as CYP6A8, 6W1, 6A2, whereas genes such as alpha esterase 6 were downregulated. The CYP6A8 genes and the others, these upregulated genes were enriched in pathways such as drug metabolism by cytochrome P450, other enzymes, glutathione metabolism of xenobiotics by cytochrome P450, and one carbon pool by folate. The downregulated genes were enriched in nucleocytoplasmic transports, lysosome, DNA replication, and others. In case of female day 30, the top upregulated genes were CYP6A8 and PCRPSC2, and GNMT, and the downregulated included some non-long coding RNA along with the unannotated genes. Like in the female day 12, day 30 also included these enriched pathways of drug metabolism by cytochrome P450 and other enzymes, as well as xenobiotic metabolism. Similar enriched pathways of downregulated genes such as lysosome, nucleocytoplasmic transport, and also the polycomb repressive complex. In female day 50, the upregulated genes included GSTE5, CYP12D1P, DGAT2, and the downregulated included LSP1 alpha, LSP1 beta. Because the number of DEX in female day 50 were so less, it was not enough for pathway enrichment. So no pathway enrichment for the genes upregulated or downregulated in female day 50. Then we plotted this heat map to see how many DEX were significant across all the ages in female. Some notable, there were total 19 significant DEX across all ages in female, and some notable ones include CYP12E1, CYP6A2, CYP6A8, GNMT, GSD1. Now, while looking at the male, we see top top upregulated gene like GST-D2, YP1, YP2. There were not many upregulated genes, hence there was no pathway enrichment. But in case of downregulated, the genes were MYB68E and some other unnotated genes that were involved in enrichment of pathways, two pathways, glycine 7 and threonine metabolism, and one carbon pool bipolar. In the male day 30, the response was quite similar to the previous female 12 and day 30, as genes such as CYP4A3, they were upregulated, which resulted in enrichment of drug metabolism by cytochrome P450 and other enzymes. The downregulated gene included COT6 and others, and they were involved in enrichment of oxidative phosphorylation, proteasome, and others. Now, oxidative phosphorylation being downregulated here is quite interesting because this pathway was upregulated in female day. In late age of male day 50, again, genes such as CYP6A8, DOP2R, YP1, they were upregulated, with downregulated like some downregulated included some long non-coding RNA. These upregulated genes in the male day 50 were involved in drug metabolism, glutathione metabolism, xenobiotic metabolism, and drug metabolism through cytochrome P450. Now, in the males, there were total 51 significant decks across all ages. Some notable ones include CYP6A2, 6A8, GST-D2, and GST-D8, among the others. Now, something that we noticed a lot, even in the previous volcano plot, as well as the pathway enrichment, was we see this drug metabolism or xenobiotic metabolism, glutathione metabolism. These metabolism being persistent across many time points in both males as well as females. So this heat map plot includes the enriched pathways that are at least enriched in two of the time points or the condition. As I said, yeah, these pathways are enriched, but we also noticed that although some genes, they were upregulated in day 50 female and day 12 male, but they still were not enough for this pathway to enrich. So we looked at them from the gene perspective, individual gene perspective. So the two genes that did come up a lot, which was CYP6A2 and CYP6A8, when we look at their expression, we see that in both males as well as females, there is a significantly higher expression in capsaicin than in the control. We suggest that capsaicin is inducing drug metabolizing genes across sexes and ages. This is again clear with this table of genes involved in xenobiotic detoxification. This table includes genes that has that includes at least one of the differentially expressed gene in any one of the time point. So we see that in one time point or the other, these genes involving xenobiotic detoxification are being upregulated significantly. And when we especially look at the expression of CYP6A2 and CYP6A8, we see them being significantly differentially expressed across all the time points. So with these evidences, we propose a hypothetical model of xenobiotic response as a route to lifespan extension. So we propose capsaicin is taken as a xenobiotic, which goes on generating RAS that activates the CNCC pathway. CNCC, it detaches from KEAP1, enters the nucleus, binds to the MAP factor, and then together go on bind to the antioxidant response element, which upregulates the drug metabolizing genes. Now, the activation of these drug metabolizing genes, or in general, xenobiotic response, leads to clearance of toxic compounds, even the endogenous toxins. And we believe that might be contributing to the longevity. The second hypothesis we are proposing is related to the expression of GNMT. So we observe GNMT being significantly upregulated in all the time points, but only in females, as males did not have such clear expression. Now, GNMT is this enzyme, glycine and methyltransferase. It is responsible for catabolizing SAM as adenosyl methionine, which is a universal methyl donor. Now, what GNMT does is it catabolizes SAM by methylating glycine into sarcosine. Now, this is important for the aging context because with age, the level of SAM increases. And GNMT is responsible for buffering that age-dependent SAM increase. So with that, we propose our second hypothesis, which is capsaicin could be activating the CNCC pathway as shown earlier, or it could also activate the deep-FOXO pathway, because of which GNMT is being expressed. Now, the GNMT enzyme, it goes on methylate glycine into sarcosine, and this whole process could lead in decline of the age-dependent SAM increase. It could also increase spermidine or glutathione, which is an antioxidant defense. These collectively or individually could help in longevity extension. Now, the third hypothesis we are proposing is related to the climbing behavior that we observed earlier. As I said earlier, we observed a significant climbing improvement in flies with capsaicin, but in the females at the late age of day 50. So I looked at the gene ontology terms of females at day 50, which showed these terms such as transmitter-gated ion channel activity, lichen-gated ion channel activity, and cholinergic synaptic signaling being enriched in these gene ontology. Looking at the CNET plot of these GO shows the genes involved in these terms. Most of the genes were anti-CHR alpha, which is an acetylcholine receptor alpha 6, 7, 1, 3, and more. We also compare the expression of those acetylcholine cholinergic receptors across female day 12, day 30, day 50, and also in males. And we see a very clear coordinated expression of those genes in female day 50, whereas not so clear in the earlier time point of female day 12 and day 30, and pretty much non-existent in the males. We had also performed a WCCN analysis for this with the help of our lab member Gerardo. And here in the WCCNA, we looked at the module-trait relationship heat map for the female's lifespan. I looked at where do these genes fall in these modules. These genes fall under the maroon module, and this maroon module showed the highest positive correlation with day 50 capsaicin female. So with even an independent WCCNA analysis showed suggested towards these cholinergic signature. Hence, even in the both level of gene and network, they both identify cholinergic signature in day 50 capsaicin female. So, in conclusion, we observed capsaicin does extend survival in both the sexes. In an organism lacking a TRPV1 ortholog, capsaicin also preserves health span by improved climbing in old females and enhances stress resistance in day 30 flies. We saw that sex was the dominant driver of the transcriptional variation. Capsaicin acts through interaction with sex and age and not as a main effect. Sustained induction of drug metabolizing enzymes such as 6A2, 6A8 was upregulated across all ages and both the sexes, possibly resulting in longevity. We also observed a female-specific GNMT-linked metabolic fluid and a late-life cholinergic signature connected to improved climbing. The hypothetical models that we proposed were based on transcriptomic profiling. So this only suggests correlation and does not establish causation. We did the whole fly transcriptomic that captures aggregate signals across tissues. So a tissue-specific profiling would resolve these signatures to their sources. Also, our study assessed a single concentration. So a dose response experiments would establish the effective brains, as well as any dose dependence. So these are the future works we are planning to do. We are planning to perform the functional analysis with the key genes that we had found, GNMT, CYP6A8, and CYP6A2. We are also planning to perform targeted metabolomic profiling for SAM, sarcosin, and spermidine that will help to confirm the GNMT route that we mentioned earlier. And we also are planning to do the biochemical assay with protein, glucose, and triglyceride, quantifying in the body. I'd like to acknowledge firstly Dr. Reddy, my advisor, for all the guidance that he has given and also giving an opportunity to grow as a researcher. My committee members, Dr. Hankins, Dr. Padma, and Dr. Mohan. Special thanks to Dr. Mohan for not just the professional help, but also the encouragement throughout. I'm also thankful to our postdocs, Dr. Rima, Dr. Carlos, and Dr. Pillai. I'm thankful to my friends and also my lab mates, Alan, Arishi, Gerardo, and all Dr. Reddy's lab members. I'm grateful for my family for everything, my dad, my mom, my sister, my brother-in-law, my nephew, and my fiance. And I'm thankful for my family away from home, Binju, and all my friends, Vinita, Ramya, Vaibhavi, Vivian, Desiree, Sarah, Rebecca, Anjan, and anybody else if I've forgotten. I'd also like to acknowledge the grant NSF and USD and IFA, without which this project might not have been possible. With that, I end my presentation with this takeaway message that let's eat peppers if we want to live longer and importantly, healthier. Thank you.